So we'll begin Chapter 1 actually on page 1 of your textbook. And what we're going to look at in this chapter are some of just the main themes of microbiology. So this is essentially just an introductory chapter. And so a lot of the stuff in here should sound familiar from maybe biology. Of course, if you have been out of biology for several semesters or even years, then you definitely want to pay attention to the vocabulary in this chapter and maybe use some of the review videos for the next couple of chapters that go back over the cell parts and things like that. They can be found in the content folder of our Blackboard course. I did just want to remind you that these are the notes for Chapter 1, and so it would be a good idea to go ahead and print these off. And so as I go through this chapter with you, you can see the objective, and then on this side you could write out the answer to it. A great thing about these videos is you can pause them, you can go back and listen to a part again just as many times as you need to. So again, starting at the very beginning of Chapter 1. And so the first thing that we look at is the name of this course. So microbiology. What is microbiology? Well, by definition, it's the study of organisms too small to be seen without magnification. You know, if you break that word down, micro means small, bio means life or living, and then logi comes from the Greek word logos, which means to study. So in this course, we're going to study living things or organisms that are microscopic. They are too small to see without using some type of magnification. And so these organisms that are so small are called microorganisms. And just to kind of give you a quick list here, these are the microorganisms that we're going to be discussing in this class. So you see bacteria, which there's a picture here. You see fungus, you see protozoans, helminths, which are worms, algae, and then viruses are listed here as well. Now, I would just kind of mention this, and you'll hear me say this in class, is that anytime we give you a list, you want to make sure that you're familiar enough with that list that you know what does not belong. So we know these are the microorganisms that we're going to look at in this class. Now, I would just go ahead and briefly mention this, and we'll pick this up again in a few moments. Viruses here, be mindful that they're technically not living things. We'll talk about why a little bit later on, but we just go ahead and list them here because they are microscopic and we are going to talk about them in our class. Okay? So with those organisms or microorganisms that are listed there, we can take them and divide them into two basic categories: prokaryotes versus eukaryotes. And again, these terms should sound familiar with biology. And so if we look at the word prokaryote, it actually comes from the words that mean pre-nucleus or before nucleus. And so we typically tell our students in biology, pro rhymes with no. So an organism that is a prokaryote, although it is made of a cell, the cell does not have a nucleus. Now, be careful. That does not mean that they don't have genetic material. They still do. They still have DNA and materials like that, but they don't have it bound in an actual nucleus. Other things we know about prokaryotes, they're very simple. They are very, very small, typically unicellular, and in addition to not having a nucleus, they also don't have all those other membrane-bound organelles that we talked about in biology. So you won't find the Golgi apparatus, you won't find mitochondria and things like that inside of them. So if you look at a basic prokaryotic cell here, and this is an image that you can view in your textbook on the bottom of page 6, but you'll just see here's the cell. It's very basic. You'll notice it does have chromosomes, so it does have genetic material, but it's not bound in a nucleus like other types of cells would be. So that's the first category, and as far as our list, if we go back, that's where you're going to find bacteria. So bacteria are located in the category of prokaryote. So if you look at the second category, the eukaryotes, this comes from the root word that means true nucleus. So the way that we always tell students to remember it is pro rhymes with no, eu rhymes with true, or they do have a nucleus. So with eukaryotes, you would find the genetic material in that membrane structure that we call the nucleus. Eukaryotic cells are typically more complex. They have a lot more parts. They can be unicellular, meaning just made of one cell, or multicellular, made of many. And in addition to the nucleus, you'll find all those membrane-bound organelle. So if we look at a eukaryotic cell, here's that membrane-bound nucleus, and then all these other organelles like the mitochondria. You'll see ribosomes here, you'll see lysosomes and vacuoles or vesicles, you'll see the rough ER and the smooth ER, all those different pieces that we talk about in biology. So if we go back to that main list, this is where you're going to find fungi, protozoans, helminths, and algae, all of those located in that eukaryotic group. And then, of course, just to briefly remind you about viruses. So viruses not found in either of those two categories. They're kind of found in their own category here because they're non-cellular. And so if you remember back in biology, the basic unit of life was a cell. So what that meant for us is in order to be a living thing, if we break you down to your smallest pieces, you've got to have cells. Well, viruses don't. So because they're non-cellular, we also consider them to be non-living. One of the things that we know about viruses, though, is that they are parasitic. They are what we call host dependent. So the only way that viruses are able to actually reproduce, because they don't have all those cell parts, is because they'll find a host cell to actually penetrate into. And from there they're able to make more of themselves. With viral structure, we'll talk about this later on in the unit, they simply just have an outer shell, they have nucleic acids on the inside, and then proteins. And that's really all there is to their basic structure. So again, here you can see simple viruses, not cells or cell parts, but just an outer shell and the nucleic acid here. But you can see there's lots of different shapes and sizes with those. Okay, so you can go into a little more detail here if you want to, if you're looking at objective number 3. We really have just covered all of this, but just to kind of reiterate, prokaryotes versus eukaryotes, who's found in those categories, and then looking at viruses as well. Okay? So I always have students ask, why do we talk about microbiology? Why is this a course that we're required to take? We're assuming that most of you are some type of health-related major. So a lot of you probably plan on going into nursing. Some of you plan on going into medical school, or just a biology, environmental science type of major. And so a couple reasons why microbes are important. One of them is roles that they play in our ecosystems. You may not be aware of this, but microbes or microorganisms are heavily involved in photosynthesis. Most of us, when we think photosynthesis, we think plants, right? And that ability for plants to take carbon dioxide, convert it into oxygen, as well as to use light sources to help make food for themselves. But plants are not the only ones that can do photosynthesis. There actually are a large amount of microorganisms that are photosynthetic. It's actually believed that microorganisms were the first organisms on the planet to actually do photosynthesis. They're the ones that actually put oxygen into the atmosphere so that plants could then develop animals and so forth and so on. So very important for photosynthetic purposes. But also, microbes are heavily involved in decomposition. If you know what that word decomposition or to decompose, it simply just means to break down dead material. And so a lot of microbes are heavily involved with recycling nutrients. Now, you may not think that that's an important concept, but if we did not have decomposers, we would have dead material just lying around and we would not be able to do anything with it. So if you look at like roadkill on the side of the road, the longer it stays there, eventually it gets broken down. Now, part of that's from, buzzards and rodents and things like that, but the other part are the bacteria and fungus that will actually develop on those organisms and break them down. You think about fruit. As fruit sits on your counter, eventually it starts looking like this. Now, obviously, you would not want to eat this, but what will happen over time is the mold or the fungus that grows on it will eventually break that fruit down completely. Now, this is important for other purposes, talk about later on. We derive penicillin from mold, so it actually can be useful in that sense as well. So some other things that we use microorganisms for, not just involved in the ecosystem, but we as humans use them a lot. One is for what we call biotechnology. So we use microbes to produce foods, drugs, and vaccines. You think about fungus, we think about mushrooms being used for food. A lot of different types of microbes are used as food additives, so food flavorings. We already mentioned penicillin. It's derived from a microorganism. We also use a lot of vaccines today to help prevent infections or to treat infections, and a lot of those vaccines are derived from the microorganism themselves. So biotechnology definitely something that we use microbes for. We also use microbes in genetic engineering, so you probably have heard of GMOs before, genetically modified organisms, and so a lot of microbes are being genetically modified to help us develop new products, one of which we think about just going to the grocery store. A lot of the foods that we eat are genetically modified, so they have a longer shelf life, but also using them to develop new products, like using microbes to develop insulin and things like that to help treat human ailments. And then another really important way that we use microorganisms is with bioremediation. Now, the difference here is we don't manipulate the organisms. We actually take them as they are, and we use them to remedy environmental problems. So, for example, oil spills. There are certain types of bacteria that can be introduced into that environment, and they absorb the oil like a sponge. Or we think about water sewage treatment plants. You know, when you flush the toilet, that water has to go somewhere to be cleaned, and so it goes to our treatment plants. And one of the ways they clean them is they use microbes to pull the fecal matter and other materials out of the water, so that what comes back through your faucet is considered to be clean. So microbes are very important. We're going to talk about a lot more of this as we go throughout the semester. But of course, other reasons why we want to talk about microbes is not just the fact that we can use them, but we also need to understand them because there is the potential that they can be dangerous. Now, I do want to mention, as far as microbes are concerned, a majority of them are free living. So a majority of microbes, bacteria and fungus and helminths, they live in free existence, relatively harmless to us. Okay? There's only a small percentage of microbes that are actually considered to be parasitic or pathogenic. And so when we talk about parasites, definition is that they live on or in another organism, and they typically harm that organism. So we think about like tapeworms, we think about infections like MRSA and things like that. Those organisms are being parasitic. And so another vocabulary word that we use with these is the term pathogen. So pathogen is any type, it's actually a disease-causing agent, but specifically for our class, it's the microbes that cause disease. Just interestingly enough, nearly 2,000 microbes cause disease, which you're thinking 2,000 is a big number, but when you think of how many microbes are on this planet, 2,000 is really only a small number, okay? But those 2,000 microbes are causing millions and billions of infections and deaths worldwide. So one of the things that we see here, and definitely something that you're going to want to make note of, is a table that's actually found in your book. So this is, it's on page 11, table 1.2, and so what they do with this is they're looking at worldwide morbidity and mortality of common diseases. So we look at reasons why worldwide people are dying, and then they've got some other charts and tables they look at. So if you look at the list, the number one infectious disease that's caused by a microbe that leads to the highest amount of deaths is influenza. Well, what's influenza? Well, that's the flu. And so, you know, a lot of people think the flu is not a big deal, but this is a very, very serious thing. But one thing that we'll note about this is that influenza is a respiratory disease. So it's pretty easy to see why it would be at the top of the list, because obviously your respiratory system is always open. So this is where we take in majority of microbes into our body. So easy to see why a respiratory disease would be the number one cause of mortality worldwide. Others, they mentioned typhoid, which is something we don't see a whole lot here. Measles, you've heard of before. And then you get into other things like HIV, Shigella, which is like a foodborne pathogen, malaria, you know, that's the one that's transmitted by mosquitoes worldwide, and then TB, definitely something that I'm sure a lot of you in the medical field have heard of before. And then just another way of seeing it from this table as well. Now, one of the things that we'll note is, as far as we are concerned, United States does a much better job in dealing with infectious diseases. When you kind of compare deaths in low-income countries versus upper and higher income countries, you'll notice that infectious diseases are a huge problem in third world countries. So those countries that don't have a lot of money, those countries that don't have a lot of access, versus high-income countries where we do have more money, we do have more access to medicine, to education, and so you'll see it being a much bigger problem in other parts of the world than it is here, although it's still a problem here, not as large a problem as in other parts of the world. So one of the things that they do talk about in Chapter 1 is a little history lesson. And the history lesson is dealing with what we call spontaneous generation. Now, I'm not going to go through the whole historical thing. You can read through this section in your text. But one of the things that I did want to mention was a theory that they used to have. So before we had medical advancements like we do now, there was an early belief in something called spontaneous generation that just simply said that life could just arise. And the idea of this came from, people would think, if they left meat out on the counter, eventually there'd be flies, right? And so they just had this idea that, boom, all of a sudden, living things just poof appeared. And what actually disproved that was, and you can, like I said, you can read back through this, but there was actually an experiment that was done. And in this experiment, they actually took meat and they put it in several different locations. So they put it out on the counter. They also put it in a jar with a lid. And what they noticed was eventually that the flies did not just appear, they had to emerge from something else, which were maggots. Maggots typically are really small, so you wouldn't see them on the meat until they actually turned into the adult form, which was the fly. And so they were able to disprove this theory. But one of the things they mentioned about microbes is it's easy to think that, boom, this stuff has just appeared because it's microscopic. But just because it's microscopic doesn't mean it just poof appeared. It was already present. And so if you will, you can read back through the conversation about that in your textbook and get a little bit better idea of what's going on. It's on page 14 in the Making Connections area. Okay. Some other vocabulary words that we talk about in this chapter are kind of getting towards the end here. The difference between emerging and re-emerging diseases. So definition of an emerging disease is a newly identified condition. It's being reported in increasing numbers. So some examples they mentioned of this, like Ebola. So Ebola virus or fever virus, it's a newly identified condition that we saw what was it, a couple years ago or last year, and they mentioned HIV is another one that's here. So emerging would just be it's newly identified. Re-emerging diseases are those that we've known about for a while, and then all of a sudden we have increasing numbers of these. Great example of this is tuberculosis, right? So tuberculosis is something that's been around for a while. And 10-15 years ago, you didn't hear a whole lot about it, right? But here recently, the last probably 5-6 years, we've been hearing a lot more about TB because it is re-emerging. It's becoming more of a problem now than it used to be. Same thing with influenza. The flu was not something that they really discussed a whole lot when I was a child. But now, once I got into college, got into career, the flu is something that is highly prominent, and you hear a lot of talk about it. And so difference between emerging and re-emerging. Emerging is totally new. Re-emerging something that we had, it seemed to kind of go away, and then now all of a sudden we've got a problem with it again. So just kind of terminology with those. All right. So if you look at the very last page of our objectives, or actually, yeah, the very last page, we kind of end the chapter with some vocabulary here and some classification. So they define the word taxonomy. So taxonomy, it's an organized way of classifying organisms. It's just a formal system that we use to organize living things. So to figure out who's related to who, what makes them not related, and just able to kind of categorize organisms. So we use classification, which is the arrangement of organisms into groups, and from there we use nomenclature to assign them names and identify them. So just some vocabulary words that are mentioned here. Now, one of the things that we deal with in biology, we'll come back to here, is that idea of taxonomy. And so if we look at taxonomy, we use levels of classification. And the way they're listed on the screen is in the hierarchy of most inclusive to least inclusive, or your largest to your smallest. So we have these listed: domain, kingdom, phylum, class, order, family, genus, species. You want to make sure you know the order. And I'm sure when you were taught this probably in high school, you were taught the phrase, "Did King Philip come over for good spaghetti?" because that's an easy way to remember the levels of classification. So when we find a species, the first thing that we do with the species is determine what domain is it found in. So is it in domain archaea, domain bacteria, or is it in domain eukarya? From there, we look at its characteristics and determine what kingdom it's found in. Once we determine its kingdom, we get a little more specific based on its characteristics and determine its phylum or division. From there, we determine its class, its order, its family, its genus, its species, and we get down to genus and species, we get to a very specific organism. So we look at some examples here. If you were to come across a species and you determine that it was in domain eukarya, so you determine it's a eukaryote because it has a true nucleus. From there you would determine, okay, well, what kingdom is it in, right? So is it an animal? Is it a plant? Is it a fungus? This example, you can see it's a protist over here. Over here, let's say that it's an animal, okay? Well, from there, you think about, there's a lot of animals in the animal kingdom. So you get a little more specific and say, okay, well, what if it's in phylum Chordata? So these are those that have a chordate. Well, you'll notice when we list those in phylum Chordata, C star doesn't fall here anymore because it's not a chordate. What if we get a little more specific and we just focus on class Mammalia? So these are just your mammals. You'll notice the sea squirt is not a mammal. Doesn't have mammary glands, doesn't have hair or fur, and so it's no longer found in class Mammalia. If you get a little more specific, look at order primates. So these are just those that have our type of vision, opposable thumbs, and so you'll notice that in primate you don't see a dog, right? We get a little more specific and look at family Hominoidea, and so for this one we'll notice that the raccoon is not here. You get a little more specific to genus and species Homo sapiens, that's just a human. And you can do the same thing for kingdom Protista, getting all the way down here to a paramecium. And so you'll notice we use these levels of taxonomy to kind of classify or categorize organisms, and you can see kind of how they're related, how closely they're related, until you get to your final genus and species. Now, we did just want to mention this, just to make certain that you were familiar, the three domains, so that top category. So you have domain bacteria. These are your prokaryotic true bacteria. You have domain archaea. Now, these are the ones that are going to basically mimic prokaryotic bacteria, but they live in extreme environments. That's what makes them different. And then you have domain eukarya, those that have a true nucleus. So just a quick check. Organisms in the same family must also be in the same class. Is that true? Remember, domain, kingdom, phylum, class, order, family, genus, species. So if they're in the same family, must they also be in the same class? Well, let's go back. In the same family, should they also be in the same class? Yes, they should. And so the answer on this one actually is true. Okay? So very last thing that we'll mention here is objective number 11. So how to write a scientific name correctly. This is something you're going to want to learn because you're going to need to do this in lab. So we use what we call binomial nomenclature, which just says living things have two names. Those two names are comprised of the genus and the species. Now, when you write these, the genus is always written with a capital letter. Species is always written with a lowercase letter. And when you write them, you either have to italicize both words or underline both words. So for example, Staph aureus. So Staphylococcus is the genus name, which is why it gets the capital letter. Aureus is species with the lowercase, and you either have to write them both in italics or underline each word. And that's the end of chapter 1.